Hey y'all, it's Miss Main here. Today we're going to talk about meiosis. Meiosis is different than mitosis. There are two divisions. We have crossing over that causes genetic variation. We will end with 4 haploid daughter cells instead of 2 that are genetically different. And meiosis occurs in our gamete or our sex cells. Just like mitosis, prior to meiosis, cells will be in interphase. There is the G1, S, and G2 phase. In the G1 phase, the cells make sure the cell is large enough for replication. In S phase, chromatin right here will actually become chromosomes. Our homologous chromosomes, one from each parent, will duplicate and become sister chromatids. So, let's say mom's chromosome is here and dad's chromosome is here. Our mom's chromosome will duplicate and become a sister chromatid. Our dad's chromosome will duplicate and also become sister chromatid. Together, they are both homologous chromosomes. Our centrosomes are also going to duplicate in interphase. In G2, the cell will make sure that there are no errors before moving into meiosis. Now we begin meiosis 1. In prophase 1, the nuclear envelope surrounding the nucleus will begin to divide, as we can see here. Homologous chromosomes will line up, matching with a tetrad. It will form a tetrad. Non-sister chromatids of the tetrad exchange genetic material known as crossing over. We can see this in this image whenever we can see right here that we have crossed and our colors have changed. Our spindle fibers from the centrioles will also attach to the tetrads at our centromere. Our next phase is metaphase 1. In this phase, the spindle fibers pull tetrads to the middle of the cell. The next phase of meiosis is anaphase 1. Here, spindle fibers pull the tetrads apart, pulling the sister chromatids to opposite ends of the cell. If you notice, you can see that there is genetic variation on those sister chromatids that are pulled to opposite ends of the cell. Our last phase of meiosis 1 is telophase 1 in cytokinesis. In telophase 1, we see a cleavage furrow begin to form. This actually splits the cell. And here is our cleavage furrow. The nuclear envelope of the nucleus reforms around the sister chromatids in both ends of the cell. Spindle fibers will also begin to dissolve. Cytokinesis means that we have 2 genetically different haploid cells. Both of these cells will now go into meiosis 2. Now we begin meiosis 2. It is very similar to mitosis. In prophase 2, the nuclear envelope will disappear in both cells. Spindle fibers from those centrioles are also going to attach to our sister chromatids at the centromere. In metaphase 2, the spindle fibers bring sister chromatids to the middle of our cell. In anaphase 2, those spindle fibers are pulling apart our sister chromatids. Now we're going to be called chromosomes. The last phase of meiosis 2 is telophase 2 in cytokinesis. We see that nuclear envelope reforming around the chromosomes in each of the cells. The cleavage furrow begins to form, splitting the cells, as we can see, and our spindle fibers dissolve. The end result of meiosis is 4 genetically different haploid cells. We can see these cells are genetically different by this different coloration on the chromosomes. Remember, this came from crossing over in prophase 1. I hope you use this video as a study aid to really study these phases of meiosis. I chose the animated images so you could see how the cell goes through each phase. Please let me know if you have any questions about meiosis. We'll do different activities in class to reinforce what we just covered here. Have a great day, Miss Main.